Welcome everybody and thanks also for joining this FcRn Symposium. And as introduced, I will present the use of the transgenic human FcRn mouse. And before presenting data which have been acquired in our laboratories or they are described in literatures, a few general considerations. A therapeutic IgG, they have unique pharmacokinetic properties. They have a half-life of approximately 19 days. They show a sustained high exposure, and they have, with that also, they have a low dosing frequency of once per week or sometimes once per month. So there are known three elimination pathways. The first one is a linear elimination pathway. It's also the endogenous pathway. That's the first step in unspecific cellular uptake, called also phagocytosis, followed by either proteolysis or the FcRn salvage pathway, and this pathway has been already presented by Greg in his presentation. The second elimination pathway, that's receptor-mediated endocytosis. That's the interaction with the pharmacological target. It's a nonlinear process, and it's also called target-mediated drug disposition, TMDD. And the last elimination pathway I'd like to mention is related to immunogenicity. As a therapeutic IgG is considered as a foreign protein for a host, then this can lead to the formation of anti-drug antibodies, so-called ADAs, and these ADAs form a complex with a therapeutic IgG, and such immune complexes are rapidly taken up by the reticuloendothelial system, also called RES. Now, why actually we're interested in the pharmacokinetics of antibodies? It's as I said, a canonical antibody or a regular antibody has these unique properties. Well, we have a paradigm shift in the discovery of antibodies. Protein engineers nowadays provide a wide range of different antibody formats, as here it is depicted in this picture below. I mean, this is a canonical antibody with a monospecific binding, but nowadays you have bispecific antibodies. You have antibodies with a fused binding moiety at the C or the N terminus, and also its combinations. So as there's a deviation from the classical canonical antibody, there is a need to select and characterize the different antibody candidates in respect to their pharmacokinetic properties. As there is no established in vitro tool currently available to do this in the drug discovery phase, animal PK studies are still warranted. And if you talk about animal PKs, mostly these are mouse PK studies. So it is known that the FcRn plays an important role in the pharmacokinetics, and there are large species differences in the FcRn interaction between the human IgG and also between the human FcRn and the murine FcRn and the human FcRn. And this is the rationale for using this transgenic human FcRn mouse model instead of wild-type mice. Now this slide here depicts actually the two major steps for linear pharmacokinetics. The one is the uptake of the antibody into the cell, and then the next step is the interaction to the FcRn in the endosome. And if the IgG is not bound to the endosome, it is sorted into the lysosome and then degraded the IgG, or if it's bound to FcRn, it's either transported out to the plasma or to its interstitium. And that's very important, or that's the key requirement now for such an animal model, is that it reflects the FcRn biology, and that means it should reflect also the expression pattern, but also the expression levels regarding FcRn. And we investigated in our laboratories the expression pattern of FcRn in various species, including the transgenic human FcRn mice, by immunohistochemistry. And you can see in these pictures various liver tissues of different species. Panel A, this is the human liver. B, that's the cynomolgus monkey liver. C and D is the liver tissue of the respective transgenic mouse lines. C is the line 32, and D is the line 276. And what you immediately see from these pictures when you compare to the panel C and D is you see the different expression levels with the different staining. The red color is the staining towards FcRn. And you see in line 276 this overexpression of FcRn, which is due to the gene construct, because here in line 276, the FcRn gene is under an ubiquitous promoter and the CMB promoter, while the line 32 has the endogenous is under the endogenous promoter. So in essence, the line 32 resembles most closely to non-human primates or to humans. Now, immunohistochemistry is a good quantitative tool to look in the different cellular subtypes in various tissues, but it's not a quantitative method. And that has been done by the group of Neubert et al. They quantified FcRn levels in various tissues of different species, including the line 32 Homo hemizygous mice, which is depicted here in this graph. What they observed is the differences, sorry, the differences in FcRn expression between the hemi and the hemizygous mice, as expected. In homogenized liver, they found FcRn levels of approximately 100 nanomolar. Now, this is very close also to the data they found in human tissue. These are really essential information for the translation from mice to humans. Noteworthy to mention that if you do a mathematical or derive mathematically an apparent concentration of FcRn in the endosome, you come up with an estimate of roughly 40 micromolar. But again, the key information here is that in the line 32, the FcRn levels in mice and humans are comparable. Now, this is a very nice investigation across the different transgenic mouse lines. This was done by a PhD student at the Roche Innovation Center in Munich, and he tested several antibodies in different mouse lines. So he tested them in the FcRn KO mice. He tested them in the line 276 hemi and homozygous mice. And he also tested them in the line 32 in the hemi and homozygous mice and compared this as well to wild type LAC6 mice. A couple of observations you can make from this graph. The first one is that the clearance values in these FcRn knockout mice they have the highest clearance values. And this is as expected, because these mice, they're not able to recycle the IgGs by FcRn. So this clearance just reflect the uptake clearance. Another observation is that the clearance values in this line 276 are in general higher than in line 32, which is counterintuitive to the data as it has been shown before with the immunostaining, because there you saw this much overexpression of FcRn. That tells me that probably the FcRn is not functional. The FcRn protein is not functional in these mice. The other observation is you see these differences in clearance values between the hemi and the homozygous mice. I can necessarily explain these differences by just stoichiometry. And the last observation is that the clearance values in this line 32 are much closest to the data which have been observed in Xenomogus and in humans. Now, this is another good example for evaluating the translatability of the line 32 mice. This is a data about this is a publication by Betts et al. And this graph shows a data analysis of 7 different antibodies in 3 different species, in humans, Xenomogus monkeys, and the line 32 mice. So the first observation just from this graph is that these mice, they have the same productivity as a Xenomogus monkey. So that's the first observation. Another observation is if you look a little bit more closer to the data. So what you see here is a plot of different clearance values. And normally you have an appropriate clearance values between 0.2 and 0.3 mils per hours per kilogram. Now, Some of these antibodies show a much higher clearance, and that's probably due to the so-called AC-SYN score. And the AC-SYN score is an indicator for self-aggregation, and with that, a surrogate for predicting atypical PK. And atypical PK is an unusual high pharmacokinetic properties, an unwanted property of a molecule. And also here, the predictivity of this line 32 is very good. So again, this all speaks in favor of using this line 32 homozygous mice. And we further evaluated this predictivity regarding the atypical or unusual high clearance values in the line 32 on our own, and we used for this 2 antibodies, and these antibodies were priakinumab and ustekinumab. They both have the target IL-23, while ustekinumab behaves very nicely, so it has some classical PK properties. Priakinumab itself has an atypical PK behavior, so it shows very high clearance in human cynomolgus monkeys and FcRn KO mice. There has been several publications about it, probably due to unfavorable FcRn properties. Certain lipophilic patches in this molecules leads to this unusual high PK clearance. If you now test these 2 antibodies in wild-type mice and the line 32, we see the following observation. The blue line or the blue dots represent ustekinumab, and you see virtually no difference between the wild-type and the transgenic animals. If you test the priakinumab, you see a substantial difference. And in line 32, the clearance is much higher. So conclusion for us is that line 32 mice reveal a more realistic prediction of the human situation than the wild type. And the reason for it is the different FcRn affinities. We have determined the KD values as a measure for the FcRn affinity for priakinumab in human FcRn and the murine FcRn, and we found a much higher affinity towards the murine FcRn. So what happens is that in the wild type mice, this molecule with the unfavorable properties is to a certain extent quote unquote rescued. And this is another very great publication which shows you a lot of data from Avery et al., where they have presented clearance data in heme and homozygous mice, line 32, and compare this to clearance data from non-human primates and into humans. And the plot here are the correlation of the in vivo clearances between the TG32 mice, homozygous mice, versus the non-human primates. So it's a very good correlation. Another very interesting aspect is when they tested different pairs of antibodies. The one antibody has a normal FcRn affinity, while the other one has an enhanced FcRn affinity. And they tested them also in both in heme and homozygous mice. And they saw also a decrease in the clearance. And we further evaluated this one with another 2 molecule. We did this with motavizumab in our labs. Motavizumab is an antibody against RSV, respiratory syncytial virus, and the wild type, as far as I know, it's already a marketed drug. So motavizumab wild type is an IgG with a normal FcRn, and there is also this YTE mutant described in literature where there's a 10-fold higher FcRn affinity. And we tested these both molecules in the line 32 in the hemizygous and in the homozygous mice. Now, here are the data when we tested these 2 molecules in the hemizygous mice. What you see is the red line. This is the motavizumab wild type, the IgG with a normal FcRn affinity. It has a faster elimination, so a higher clearance than, as expected, than the motavizumab with enhanced FcRn, the motavizumab YTE mutant. When we did the same experiment in the homozygous mice, we see also the same trend that the wild type IgG has a higher clearance than the YTE mutant, but the extent of decrease was different between the heme and the homozygous mice. The YTE mutant always showed the same clearance values in the hemizygous mice and the homozygous mice. And we further looked into the data when we compared it to available literature data. So the data has then been shown here now. Before, these were the declearance data of mogamulizumab and mogamulizumab YTE in the two different mouse in the hemizygous and the homozygous mice. These are the clearance values from the cynomolgus and in the humans. And in humans, it has been reported a roughly 10-fold lower clearance and roughly 10-fold lower clearance in humans. And that also relates or correlates very nicely to the much longer half-life. In cynomolgus monkey, quite a similar picture, not as maybe as prominent as in humans, but still a factor of 5. When we now analyze the data we had been derived, we didn't saw this huge change in clearance. That was not even a 2-fold difference, and that was maybe a 2.5-fold difference. And we speculate about the reasons of our findings here. One potential cause, what we excluded it immediately, is that, I mean, these transgenic mice, you have a chimeric heterodimer FcRn. So it consists out of murine beta-2 microglobulin and the human FcRn alpha chain. I've not seen any data so far showing that you have the same affinities between the chimeric heterodimer and the full heterodimer, but we excluded this expression. For us, it's more likely it has to do with the low endogenous murine IgG levels in these mice. And here are a few more data about it. This is these are the data of an endogenous murine IgG level. So it's a sum of the different IgGs in the mouse. So it consists of IgG1, IgG2, A, B, etc. And this is the amount of total IgG here in the wild type and the black 6 mice versus the line to 276 and 32. And what you see is approximately a 10-fold lower. So only 10% of the endogenous murine IgG levels are circulating in these transgenic mice. And the reason for it is that the murine IgG have a much lower affinity or only a weak interaction with the human FcRn. So these mice cannot necessarily recycle their produced IgGs. What is the consequence of this behavior? I mean, here is further insight in this one. That's a publication from Dr. Waldman from the 70s. And that showed very clearly the dependency of total IgG levels in the serum versus the half-life of an IgG tracer. So if the total here, as depicted here, is the total IgG concentrations in increase, the half-life gets shorter, while the serum concentration is low, then the half-life gets much, much longer. And the reason for it is the competition of the endogenous IgGs with the FcRn recycling. And we further evaluated this one as well. And for that, we used two different tool antibodies. The tool antibodies have no biological binding. And the one tool antibody had a normal FcRn affinity, and the other one had an FcRn enhanced affinity. We took these two tool antibodies and tested them in the line 32 homozygous mice. And what we found is that the PK profile was more or less identical between the two antibodies. I'll have to say that our tool antibody here has a very favorable pharmacokinetic properties. Probably due to very favorable FcRn properties. And so the addition or the mutation at the FcRn doesn't add much doesn't really improve the pharmacokinetics. That's our explanation why we have these findings. Now, if you repeated this experiment, the same experiments, but in contrast to the first experiment, we added human IgGs, so-called IVIG fraction. And we did this before the IV administration and 1 week after the IV administration of the tool antibodies. So with that dose of twice 250 mg per kg, we reached a concentration in these mice of 2 to 3 milligram per ml. That's roughly 13 to 20 micromolar concentration. And that is at least close to the steady state concentration in humans. And the result, what we observed, is that the clearance value for the YTE mutant doesn't change, but again, we have a twofold increase in clearance for the wild-type IgGs. Our conclusion out of this experiment is that the presence of endogenous IgG is relevant for the translatability of Fc engineered variants. Now, last point I'd like to make is about the effect of immunogenicity to these transgenic mice. As I said in the beginning, it's a foreign protein for the mice, and it always can provoke an immune response, leading to the formation of anti-drug antibodies, ADAs. That leads to a rapid elimination of the drug. It's generally observed in all preclinical species. It's not predictive for human immunogenicity. The immune response in these line 32 is not very prominent. That's in contrast to the data here I show, where we see the sudden loss. So very often we see subtle differences in the profile of 1 or 2 animals. So it's very difficult from the PK profile to identify this in the line 32, but also in other of these transgenic mice. It's also very difficult to detect due to the low ADA titers. Potential solution is to switch to a SCID background instead of using the black cells. Now, let me briefly conclude on that. So, the pharmacokinetics in this transgenic human FcRn mouse, line 32, is a very important assay for the characterization of the PK properties. It's very important to use this in drug discovery to identify early liabilities and to direct the optimization of the Fc modification. Expression of FcRn, and I mean with that expression levels, but also expression pattern for the line 32 is comparable to non-human primates in humans. The mouse model is as predicted as in non-human primates for linear PK that makes PK studies redundant in non-human primates. Level of endogenous IgGs has an impact on pharmacokinetics. Co-administration of human IgG, as these fractionated human IgGs, is important, but the drawback is it interferes with the drug discovery bioanalysis. Now, these assays are based on ELISA, and these ELISAs are based on detecting human IgGs. The best dosing regimen and dose strength has to be optimized to really make this into a translational model. We observe effects on immunogenicity. We observed effects on PK by the immunogenicity in these lines, and a suggestion is to use SCID mice. Please of note is that they also have very low circulating urine IgG levels. A last point I really like to make is we really need to further advance in vitro and in silico tools for the prediction of PK properties of biologics or of antibodies. Such an approach has been already established for chemical entities very successful. The human FcRn mouse model is a key asset to further advance this approach. At the end, I really like to acknowledge my co-workers, in particular Thomas Emmerich and his co-worker Andreas Wolfert, and also Claudiason and her co-workers for conducting all the PK studies that couldn't be the work of a single contributor. Then Tillman is our protein engineer and a very specialist for FcRn biology. And then last but not least, I really appreciate the support by Wolfgang and for his scientific advice. Thanks. Now I'm at the end of my presentation. Thanks a lot for joining this FcRn seminar, and I'm happy to take your questions now.